to Science Excel, where our motto is to enhance the learning curve. In today's tutorial, let's start with mitosis. Haven't we all wondered about how growth occurs in plants and human beings? And how a wound miraculously heals? Or how using yeast makes producing bread possible? Well, let me explain. There is a common factor to these occurrences and it's called mitosis. What is mitosis? Mitosis is a type of cell division in which a single cell divides to produce two identical daughter cells. So what is the purpose of mitosis? Mitosis is used to create new cells for growth, repair and reproduction. Before we dwell into the actual process of mitosis, I would urge you to visit our previous tutorial called Introduction to Mitosis, which elaborates the purpose and functions of the key organelles involved in the process. You would be amazed to know when cells undergo mitosis, they don't just divide their DNA at random for the two daughter cells. Instead, they split up their duplicated chromosome in a series of well-orchestrated steps. Let's understand this process. There are two stages of mitosis: karyokinesis, which is the nuclear division, and cytokinesis, which is the cytoplasmic division. Karyokinesis is further divided into four phases: prophase, metaphase, anaphase, and telophase. You can remember the order of the phases with the acronym PMAT. Let's start by looking at a cell right before it begins mitosis. This cell is in interphase, the late G2 phase. The centrosomes get duplicated in this stage and the chromosomes in the nucleus each consist of two connected copies called sister chromatids. In early prophase, the mitotic spindle or spindle fiber begins to form and the nucleolus disappears. The chromosomes start to condense, making them easier to pull apart later on. In late prophase, the duplicated centrosomes move towards the opposite poles and the chromosomes now are fully condensed. The nuclear membrane breaks down, releasing the chromosomes. The spindle fiber grows more and starts to capture the chromosomes. In metaphase, the nuclear membrane now disappears completely and the chromosomes are lined up at the middle of the cell called the metaphase plate, ready to divide. The spindle fibers are formed from the polar region and they attach themselves to the centromere of each chromosome. In anaphase, the spindle fibers pull apart the chromosomes, separating the sister chromatids from the middle of the cell to the opposite poles. One set of chromosomes reach each pole by the end of anaphase. In telophase, the spindle fibers are completely lost. The nuclear membrane and nucleolus reappears. The thickened chromosomes decondense and return to their stringy form. Now the cell begins to split into two. In the concluding stage called cytokinesis, the cytoplasm divides to form two new cells, each with a complete set of chromosomes identical to those of the mother cell. We encourage you to explore the summary created as a brain-friendly mind map which provides a quick insight into what we have learned. The following assessment is the key to gauge what we have imbibed. Kudos to you if you have managed to answer all. If not, revisiting the tutorial will help reinforce concepts. Thanks for watching. If you find the content of the video useful, please like, subscribe and turn on the notification bell to receive updates on new videos.